It's 12:30. So we don't have a big group yet, but let's get started. Anyway, I just want to do two things today. One is go over test 2, which was a long time ago, but we have had other things to do in the meantime. And several of you have asked if we can go over the answers to the questions on test 2. The other thing, I'm predicting that we'll have a quiz 7 on November 17th. That would be a week from Wednesday. So we have a lot of new material to cover before that quiz. Okay, if there's no questions yet, I'll share my screen just a minute. My computer says I'm sharing my screen with you. Do you agree?
Yes, I agree.
Yes.
Yes.
So here's sort of a rough representation of test 2. I'll go over the questions one at a time. First one is about coronavirus and how it gets into a cell. So you were given 4 choices of the sequence of events. And the correct one is that first the virus attaches. After that, the membrane of the host cell invaginates, and then it closes on itself to make a vesicle, which then enters the cell containing the virus. So most of you got this one right. The second question is about osmosis, membrane permeability. And before going into this, let me remind you that anytime you have a question on this topic, refer to this guide to osmosis and tonicity, which I've gone over several times, but if you adhere to these rules, you'll get it right every time. So the first point is that osmosis only refers to water. Tonicity only refers to solute. And that solutions, I should say substances, either the solvent or the solute, will always move from high concentration to low. And a solution always has 2 components, a solute and a solvent. And when you add them together, it always comes to 100%. So these are the rules. And if a question on this, and it will come up if nowhere else on the final exam, use these rules and you'll always get it right. So in the first case here, there's 2 beakers connected by a tube, and there's a membrane in the tube that's permeable only to water. Water can go through, solutes cannot. So the left beaker has 50 milliliters of 10% sucrose, and the right has 1% sucrose. And so you let this stand overnight, and then you're asked which of the following statements is true. Well, you know immediately that A and C cannot be true. because that would require the passage of solute through the membrane. And the question has already told us that the membrane is permeable only to water. So A and C are out. So then on which side has the level of the solution changed? Well, that really is asking the question, which way is water moving? Well, water is going to move from high concentration to low. So I always find it easier to draw a picture to help me visualize what's happening. But we know that the water concentration in the right beaker is 99%, whereas in the left beaker, it's 90%. So going from high to low, water will move from right to left. So that means the liquid level in the left beaker has gone up, and the right beaker, it's gone down. Does everybody see how you use the rules? Yeah. And you use the rules again for the next question, which refers to the previous one. The tonicity of the right beaker is what relative to the tonicity in the left beaker? So tonicity refers to solute. So in which beaker, the right beaker, the solute is low compared to the left beaker. So that means the right beaker is hypotonic, and conversely, the left beaker is hypertonic. That's because the solute concentration in the right beaker is only 1%, whereas in the left beaker it's 10%. And it's going to change, it's going to remain that way because this membrane is not permeable to either of these solutes. Okay, is that okay with everyone? All right, the next question is active transport moves ions across membranes against their concentration gradients. Due to electrical charge on ions, there is always an unequal distribution of ions across a membrane, and that imposes a second gradient. And what is it called? The answer is electrochemical. The next question, match the components of the plasma membrane on the left with the role each one plays in membrane function. And so I've color-coded this. Phospholipids form a bilayer. Transmembrane proteins, these are the ones that go all the way through the membrane. Now, the answer I was looking for was they facilitate passage of small molecules. But some of you answered that it blocks the transport of molecules. And in fact, that's not untrue. It's not the best answer, but I gave credit for that. You said it blocks transport of molecules. Okay. Cholesterol is a buffer. Peripheral membrane proteins are attached reversibly. And the glycocalyx is a sticky coating on the surface. So those are the answers to the matching question. The next question is about CRISPR, and I wanted just to tell me in a few words why CRISPR is such a powerful tool. And in a few words, it is site-specific. That means it targets one gene specifically. And it's universal, which means it works in any organism, from bacteria to humans. The next one is relative to cystic fibrosis. It's about the pancreas and gallbladder. They collaborate to digest food. And then I have some fill in the blanks. The acinar cells produce digestive enzymes. And there were several different answers that I gave credit for here. A major contribution of the gallbladder is to produce bile, which contains amphipathic molecules to emulsify fats. And again, anything that represented any of these answers, you should have gotten full credit for, even though the speed grader might mark you wrong. And the next question is about terms having to do with Mendelian genetics. So what is a cross? Breeding of two plants or animals. What is the parental generation? It's the original individuals that are crossed. The F1 is a progeny of a cross. The F2 is a progeny of a cross between two F1s. Phenotype is visible characteristics. Genotype is symbolic. A back cross is a mating of a progeny to its parent. A test cross is a mating to a homozygous recessive. Inbred, all loci are homozygous. And finally, an allele is the alternate form of a gene. Then we have a couple of Mendelian genetics problems. First is about Mr. and Mrs. Smith. They both have widow's peaks, which is a dominant trait, we're told. Their first child has a widow's peak, but the second one doesn't. And Mr. Smith is raised an eyebrow about that. He wonders about Mrs. Smith. Is his accusation justified? Why or why not? Well, to think about answering this, the best way to approach it is to do a Punnett square. But first you have to have a gene symbol. So how can we assign a gene symbol? I've used the symbol W here, but we know that both Mr. and Mrs. Smith have widow's peaks, and that trait is dominant. That means that each of them has to have at least one dominant allele. And so they can be either both homozygous dominant, in which case there's no question that none of their children could have a widow's peak, or one of them could be homozygous dominant and the other one a heterozygote. And so I've shown that here on the bottom Punnett square. If one of them is homozygous dominant, big W, the other one is a heterozygote, big W, little w, all of the kids would have widow's peaks because they'd all be either homozygous dominant or heterozygotes. However, if they were both heterozygous, then you see the other Punnett square, big W crossed with big W, little w, and you can see immediately that 25% of their children would be homozygous recessive, and that means they would not have a widow peak. So to answer the question, no, you would need to go through this exercise and figure out. So the answer is no, and then why not? It's because both he and his wife might both be heterozygous. And the next question, is there a question? The next one is about Earl and his wife, Emma. Earl has normal color vision. His wife is colorblind, and color blindness is an X-linked, in other words, sex-linked trait. And the normal allele is dominant. So we know that Earl, being a male, has only one X, and this is an X-linked trait. So his X is wild type. and his other chromosome is Y, has nothing to do with color blindness. His wife is colorblind, and we know that she has two X's, which I've called here X star. And so if you do a Punnett square, you can see which progeny are male and which are female. And you can see that all the girls will be carriers, but not colorblind, and all the boys will be colorblind. So that's the answer. Any question about about the reasoning, the rationale for coming to this conclusion? Okay. And the next question is also about not only Mendelian genetics, but Mendel himself. He was the first to demonstrate the existence of genes. What did he do? What were the key things he did that no one prior to him had ever done? The two things are he first inbred each trait before doing crosses. And secondly, he evaluated the F2 generation. So these are the only answers that I would accept. Some of you put down many other things that he did, but none of them was key to his success. It was these two things that allowed him to deduce that genes exist. And then we go to CFTR. You're all very familiar with that by now. And this is the pathway of CFTR from the nucleus to the plasma membrane. So to fill in the blanks, instructions for CFTR are encoded in DNA found in the nucleus. The DNA is transcribed to RNA, which then passes through a pore. And I accepted many different answers here as long as it reflected some kind of an opening, into the endoplasmic reticulum, where it is translated into protein by ribosomes. Then it's decorated in the Golgi apparatus, and then transported through a series of vesicles to the plasma membrane. And the next one, nobody got this entirely right. There are 15 terms here. And what I'm asking you is to tell me if it's associated with mitosis or meiosis or both or neither. So the first one cell becomes two cells. That is a mitosis. Remember, mitosis is simply a cell division. It's a somatic cell division where you get a precise reproduction of the genome. Second term is haploid. Well, that is B, meiosis. Meiosis occurs, when meiosis occurs, haploid cells result. Tetrads form, that's also B, meiosis. Do not get tetrads in mitosis. DNA is replicated. Well, it's replicated in both, so that would be C. Binary fission occurs, that's D, neither. Binary fission occurs in prokaryotes, not in eukaryotes. 4 cells result, B, meiosis. Random assortment of chromosomes. Oh, here I accepted either B or C. My first choice is B, random assortment occurs in meiosis, but you could also argue that it occurs in both. A diploid is A, mitosis. Homologous chromosomes pair, that's B, pair only in meiosis. Nucleoid forms, D, neither, so that's in prokaryotes. Genetically identical cells is A, mitosis. Organelles are lacking, D, it's neither. Metaphase occurs in both. Genetic variation occurs only in B, meiosis, and produces somatic cells is a mitosis. As I said, nobody got all of these right. So does anybody have any questions about?
I have something that I noticed, which is, well, on this one, I have a 12 out of 15. So I assume each of these is worth 15 points, but number 1 and 2 are swapped. So on the test, it's number 1 is haploid and number 2 is 1 cell becomes 2 cells. But on yours, number 1 is 1 cell becomes two cells, and haploid is the second one. And I noticed the only one I actually got wrong was I think number 8.
Okay, so on the test, it's reversed.
Yeah, on the test, it goes haploid first and 1 cell becomes 2 cells second, but on your answer sheet right there, it's the opposite of that. And I think that actually you marked those as being wrong because it is the opposite, and maybe you didn't notice it, or it's
Yeah,
whatever.
no, I didn't notice that, but I'll go back and correct that.
Yeah, it's the same for me too. I got 12 out of 15. And just like Anthony said, it is swap. I just
And also,
realized
yeah,
that.
me too. Same thing. I was wondering why they are wrong.
Okay, well, so many of you agree, it must be the case. Anyway, I'll go back and I'll correct that.
Awesome. Thank you.
Okay, well, thanks for telling me. Okay, the next one is about dynein, motor protein. It transports cargo from one cellular location to another. Which statements are true? Chromosomes are one of the cargoes. That's true. Dynein is active in interphase. That is untrue. It's active in anaphase. You can eliminate it with site-specific mutation. Without dynein, cell division accelerates. That's untrue. Cell division actually slows down very, very much. Dynein activity is most abundant or apparent during anaphase, true. And it's active in the nucleolus, not at all. Okay, next we have an opportunity to apply the rules of tonicity again. In this case, we have 2 beakers connected at their bottoms containing a membrane that's permeable to everything, to water and to both solutes A and B. So if you put 40 grams of solute A in the left beaker and 20 grams of solute B in the right beaker and allow them to sit overnight. And then the first question is, what is the solute concentration in the left beaker? Well, you start out with 40 grams and you leave it overnight, and this membrane is not a barrier to the solute. So overnight it's going to equilibrate and go to 20. At the same time, in the right beaker, this is going to equilibrate, and so it starts with 20 milligrams or 20 grams, and it's going to go to 10, but it contributes 10 to the other side. So 10 goes to 20 plus 10 more is 30. And the right beaker, 20 goes to 10, but it's 20 from the left beaker, so it's also 30. So the answers to questions 1 and 2 are both 30%. Then at the start of the experiment, as before you allow any equilibration, which beaker is hypotonic, so it has a low solute concentration. that would be the right beaker. It's 20 versus 40. And the other one is left. And then after sitting overnight, what are the tonicities? Would be isotonic, because the beakers have, the contents of the beakers have equilibrated. And then the last two questions. You put a celery stalk, the cut end of a celery stalk, in a beaker of water. What would happen to it? Well, it would, some of you said swell or become turgid. Anything indicating that was acceptable. And then what would be the tonicity of the celery cells with respect to water? Hypertonic. It'd have a much higher solute concentration than water has, which has none. Then if instead of water, you put the cut end in a beaker containing 2% salt, what would happen? Well, it would wilt. Water would move out of the celery into the beaker. And what would be the tonicity with respect to the beaker? It would be hypotonic. The tonicity of the celery would be, the celery would be about 0.9% solute, and the beaker is 2%, so it would be hypotonic. Any more questions or any more corrections on the test? I should mention that we will have one more test, and I'm proposing that we have it on the last regularly scheduled class meeting, which would be December 8th, a Wednesday. And the reason I'm suggesting that as a scheduling is that you could then study for both test 3 and the final at the same time. Because for the final exam, it would be the next week. And it will cover the entire course, whereas test 3 will just cover everything since test 2. So I've penciled in for the final exam, December 15th, which would be a Wednesday, would be in the middle of finals week. If one or more of you has a problem with that date, for me, it doesn't matter. The final exam can be any day of finals week. It'll be a 2
Professor, this is Alicia Carroll.
Uh-huh.
How many questions will the final exam be?
Now, I don't know. I haven't even thought about it yet.
Okay, will you be giving us a study guide?
I'll give you something, not sure what.
Okay, thank you.
But the final exam will be twice as long as the other tests. So instead of 75 minutes, it'll be 150 minutes allowed with two attempts. And so I'm predicting that it'll be roughly twice as long. In other words, if a test is about 20 questions, the final would be about 40.
How many points is the final exam worth?
200. Yeah, so it's a lot of points. It's worth preparing for. Okay, if nothing else, I'm going to stop sharing. Take away the test. And go to the next topic. It's actually, we're finishing up a topic that we started previously. So I'm going to share my screen again with you. I hope you can see this. Mendelian genetics and molecular genetics. Anyone cannot see it? Okay, I'm assuming you all can. So I've got on this slide Mendelian genetics and molecular genetics. The essential difference, or one way that you can think about the difference, is that in Mendelian genetics, genes are abstractions. So we get our ideas about Mendelian genetics directly from Mendel. Well, what did he do? He did inbreeding and he did F2s. And what did that tell him? It told him that traits like flower color can be controlled by single entities. And these entities are called genes. But that's all we know from Mendel's work, is that single entities, and the reason he concluded they're single is that there was no smearing of traits. In other words, if he crossed a white flower and a purple flower, he got just one color and no variations of that. If he went to an F2, he would get just two colors, and they were discrete colors. There was no variation. And so because of this one-to-one association of a trait, a phenotype, and a genotype, he postulated genes. He didn't call them genes, that term came later. But for him, he had no choice. A gene was just an abstraction. Molecular genetics, genes are real because we can identify them, we can isolate them in a test tube, we can manipulate them. They're discrete molecules of DNA, which is very powerful. And my goal here today is to let you know just how powerful molecular genetics is. I've chosen just a few topics that you should know about so that you can read newspapers or follow other media outlets and know what they're talking about. So I'm going to talk about, I've already talked about restriction endonucleases, review that briefly. Polymerase chain reaction, PCR, gene therapy, genetically modified organisms, and I won't dwell on CRISPR because we already have, but don't forget that CRISPR is among the most powerful of the new technologies. Okay, this first part is a review. We went over this before. The discovery of restriction endonucleases. An important point for you to have in the back of your mind is that without these enzymes, there would be no molecular genetics. You absolutely have to have these site-specific enzymes in order to manipulate DNA in a test tube. And remember that the nucleases are unusual among the, in fact, they're unique among the depolymerases. And by that I mean enzymes that take polymers like carbohydrates, fats, and proteins. There are enzymes that will take each of these polymers apart, but they're non-specific. In other words, they go in and just tear the molecule apart. Nucleases can also do that, but some of them don't. Some of them are highly specific for where they cut and how they cut. This just illustrates what I've already said. A protease can chop in the middle of a polypeptide, or it can chew off one end or the other end. but the sites where it cuts are non-specific. Similarly, lipases digest fats, so they'll take a fatty acid, which is a fat, and convert it into free fatty acids, monomers. And similarly, carbohydrates, it breaks down carbohydrates, which are monomers of sugars, and it can cut anywhere in the chain or at either end, but entirely non-specific. Nucleases, on the other hand, can be either specific or non-specific. So the non-specific ones can randomly hydrolyze the phosphodiester bonds in a chain, or they can chew off either end, either the 5' or the 3' end. Well, these, for molecular biological purposes, are of no interest. The ones that are interesting are the specific ones, and they're called restriction endonucleases. They come from bacteria, and they cut at a particular nucleotide sequence. So they recognize that sequence and they cut the DNA at the same sequence. And that is very surprising. There are 5 different types of site-specific endonucleases. We're only interested in one type, type 2. And that's because they recognize and cut at the same site. I went over how restriction and type 2 restriction endonucleases are named. Comes from the names of abbreviated names of the bacteria that produce them. Remember that restriction enzyme sites are palindromes. That means they read the same forward and backward. We talked about plasmids, these little circles of DNA. Usually they're circles. And why are they important? For cloning purposes. If you want to clone a particular piece of DNA or a library of DNA fragments, you need a plasmid. And I went over all this in detail in the previous lecture. So to review for the test, go back to their video recording of that lecture where I described these things in detail. This is how you clone into a plasmid vector. And in this case, this is how you make a library of DNA in a plasmid vector. And essential for many of these processes is an enzyme called reverse transcriptase, which converts RNA to DNA. Now, uses of restriction enzymes, there's one type of use that I went over before, but I'd like to emphasize this because you are exposed to it on a fairly frequent basis. You hear about DNA evidence. It's used in trials as evidence in court, and that is the most powerful evidence available because it's unambiguous and it gets it right every time. I went through in the previous lecture how polyacrylamide gel is set up and how it works. You get these digests of DNA which come out as bands. And then I gave you an example of its use in forensics, where there was a rape case. So you have DNA from the victim. You have DNA from 3 different suspects, A, B, and C. And you have DNA that was collected from the site of the rape. So all of these DNAs were digested with different enzymes, run on a polyacrylamide gel, so they're separated. And the pattern is different for each one. So the victim has a pattern. The three different suspects each have a different pattern. And the evidence from the site of the crime has a particular pattern, and it matches exactly with suspect A. So this is unequivocal evidence that suspect A did the crime. And I think that there is no court in the country that does not accept DNA evidence. So when you hear about DNA evidence, now you know how a lab gets that evidence. So that was a review of what we'd gone over before. Now I'm going to move on to the second topic, which is polymerase chain reaction. Oh, and before I start it, I want to show you a video.
Thanks for stopping by. This is 2 Minute Classroom, and
Can you see this?
Yes.
Yep.
Okay.
today we're talking about polymerase chain reaction, or PCR as it's more commonly known. First, let's talk about what PCR is and how it works, and then I'll give some examples of what PCR is used for. PCR is a technique used to amplify or copy small specific segments of DNA. For PCR to be successful, you need the DNA sample you want copied, DNA primers to identify the specific sequence of DNA you want to amplify, lots of raw nucleotides to form the DNA copies, and a heat-resistant DNA polymerase to make the new copies. This all goes into a special stable solution. Once all the ingredients are in, you run the solution through many temperature cycles until you reach the amount of DNA copies needed. There are 3 steps to this cycle. Step 1 is denaturation. The solution is heated, allowing complementary DNA strands to separate so that all the DNA in solution is single-stranded. Step 2 is annealing. The solution is cooled so that the DNA primers can bind to the specific region of DNA you are interested in. Step 3 is extension. The temperature is raised so that the DNA polymerase can bind and perform the necessary operation of synthesizing new DNA strands. This cycle is repeated about 30 times, takes about 3 hours, and creates over 1 billion copies. So what is PCR actually used for? PCR has many research-based and practical applications. It's used for DNA mapping, like in the Human Genome Project. It's used to check sections of DNA for genetic testing to determine potential disorders. Companies like 23andMe and Ancestry.com use PCR to run personal DNA tests. And my favorite application for PCR is in forensics. Whenever a crime is committed, the perpetrator almost always leaves their DNA at the crime scene. This could be hair, semen, skin, or even blood, for example. Investigators search for this evidence and, thanks to PCR, can amplify the DNA and compare it to the DNA of suspects. This genetic information is then put into the FBI's DNA database, CODIS. But it gets even better. Investigators have been able to take DNA evidence from crimes committed long before CODIS was put together and run it through the current system. This has solved many cold cases and also exonerated prisoners who have been wrongfully accused, even some that were on death row. Needless to say, PCR is one of the most important scientific advances ever, and now you know why. If you have additional questions, I'll do my best to answer those.
Back to the slide, polymerase chain reaction. Anybody can't see that? Okay, I'll assume you can. That's video. Now, I'd like to go over stepwise what you just saw in the video. It's really important that you understand how polymerase chain reaction works. If you just look at this flow diagram, where it starts at the top and says target sequence. So this is a piece of DNA. It could come from a genome, it could come from a crime scene, but it's a single molecule. First step is you heat it. Well, I should say that actually the first step is that you put this DNA sample, and it could be the whole genome of an organism, into a little tube which contains primers specific for the target that you're aiming for, the triphosphate bases, the A's and T's and G's and C's, and a very special enzyme. So you have this mixture of 4 components. The first step is called denaturation, where you heat this little tube up to almost boiling, to about 95 degrees, which causes the strands of this target to separate. Then the next step is annealing, which means you cool it down to a much cooler temperature, about 50 degrees. And that allows these primers to attach to the target, that they're complementary sequences. Then the temperature is raised. The third step is the temperature goes up to about 75, which is the temperature at which this very special enzyme works. And so now, and so this enzyme, it's a DNA polymerase, it fills in the gaps. All the A's and T's and G's and C's are filled in. So after one cycle of temperature switching, this one molecule of target sequence is now 2. And then you do it all over again. You heat, cool, extend with a moderate temperature, and now these 2 copies have become 4. And then you do it again. Cycle 3, now the 1 copy has gone to 8. And after cycle 5, there are 32 copies. After cycle 15, there are 32,000 copies. And after cycle 25, there are 33 million copies, all starting with a single copy. And so you've gone from 1 copy of DNA to millions of copies in just a couple of hours. And it's very,
Professor.
yes.
This is Steve Stinson.
Uh-huh.
You do not have to add any more material once you start the process. It's just, once you've added what's in there, it will automatically do this multiplication.
That's right. And that's what you mentioned. It's just a beautiful thing about this process. You mix up, you see these pictures of little tubes here on the right. You take a little tube like that, and you have a solution with just these three components, the primers, the triphosphate bases, and this special enzyme. Into that solution, you put your target sequence that you want to amplify. And it can be as little as one. It can be more than that, but you have to have at least one sequence. And then by just continually switching the temperatures, you end up with millions.
I have a question.
Yeah,
Is this process similar to mitosis?
no,
Not at
No.
all.
Not at all.
Okay.
This mitosis occurs in a cell. And this has nothing to do with the cell. You start with raw DNA, which came from a cell, but either you find it somewhere or you break up some cells to get it. And then you do this process, temperature cycling. So it's beautiful in its simplicity and its power, where you need to start with only a single molecule and can make as much as you want.
This process keeps it from making any genetic mistakes. It's like perfect in a sense. There's no room for error.
Yeah, it is very a very high fidelity. Yeah, and this process PCR goes on every day in labs around the world, and it never makes mistakes. If it's done properly, it does not make mistakes.
And no matter if the cell, if the forensic that they get is dead or alive, it will still work.
Yeah. So if we go back to this this slide about forensics, this evidence from the site could, and you remember from the video, that it could be a blood sample or semen or a hair from the site. But if there's one molecule of DNA from the crime scene, that can now be amplified with PCR to as much as you want. And once it's amplified, so you have a test tube full of DNA, it can now be digested with restriction enzymes, so you get these diagnostic bands. So you can see how the different technologies work together to solve a problem. We have with the restriction enzymes, we have PCR, we have polyacrylamide gel electrophoresis, and that provides, in the case of forensics, DNA evidence. So just to give you a sense of how you do this, these reactions are always run in what are called multi-well plates. In this case, there are it's a plate with a particular footprint. It has 384 little holes in it. Each hole is the size for one of these little tubes. And these tubes can be filled with an individual pipetter like this. At some point, you'll need a little centrifuge. Or there can be automatic pipetters, which will do as much as a whole plate at once. And then the plate is put in a thermocycler like this. This is a picture of a modern thermocycler, or they can be operated by robots for doing very large numbers. And just for historical purposes, back in the 1980s, when Taq polymerase, the DNA polymerase was first discovered and PCR was described, we did it with water baths. So we'd have a water bath with 3 different temperatures of water. So you'd mix up little tubes in a plate like this, hold it at 94 degrees centigrade for a few seconds, then put it at 55 for a few seconds, then put it at 75 for a few minutes. That's cycle 1. And you take it back to the 94, and you do that half a day or more, depending on how efficiently the reactions were working. And but it was very boring. And the reason people did it is because it's so powerful. It really works. Nobody does it like this anymore. Anybody that does PCR has an automatic thermocycler. Now, how did PCR get discovered? It's a really interesting history. Before PCR, which means before the 1980s, the only way you could get DNA is to isolate it from a living organism. So, for example, if you wanted to get bacterial DNA, you could break up E. coli cells, get the DNA out. If you wanted to do with humans, you do the same thing with human cells, but you had to start with cells. Then in 1969, a man named Thomas Brock isolated a new species of bacterium from a hot spring. Now, hot springs are at about the temperature of boiling water. So if there's a bacterium living in a hot spring, it means that its enzymes are not denatured by heat. It can live in temperatures up to the point of boiling. Well, the bacterium that he isolated was called Thermus aquaticus. And many enzymes have been isolated from that bacterium. They're all heat tolerant, but one of them is a DNA polymerase called Taq, T-A-Q, Taq polymerase. And that's what's used in PCR. But isolating this enzyme or this bacterium and the enzyme from it didn't really predict anything. But in 1979, there was a biotech startup in San Francisco called Cetus Corporation. It hired a man named Kerry Mullis to do a particular job, but it didn't really monitor him closely. So he went off and did what he felt like. And what he ended up doing was discovering PCR. So in 1986, he made a presentation at a scientific meeting, and he published this idea of a chain reaction. An interesting side note here, he got a reprimand for the Atomic Energy Agency for publishing about a chain reaction without permission. Of course, what they thought, it was a nuclear reaction, not a molecular reaction. Well, in 1993, Mullis was awarded the Nobel Prize for this because immediately, as soon as he published it, everybody could see the power of it, and labs all over the world immediately started using it, even though they had to do it in water baths at that time, not thermocyclers. Another interesting side note here, Mullis goes to Stockholm and gives his Nobel speech. And in the speech, he pointed out that he really didn't have any joy the night he proved that PCR would work, because his girlfriend left him that night because he spent all his time in the lab and ignored her. And a final interesting point is that the Cetus company gave Mullis a bonus for his discovery, $10,000. they turned around, excuse me, and sold the technology to a pharmaceutical company, Roche, for $330 million.
Did he get the girlfriend back after he got all that money?
I don't, yeah, that's a good point. And I don't know the answer, but you'd think she would have come running back.
Oh, you mentioned the hot springs. That's those hot water coming out of the ground from
Yeah.
volcanic.
Yeah,
Okay.
if you've been to Yellowstone Park, you probably have all seen the little bubbly, they're smoking hot and bubbling sulfur compounds. They smell bad. Anyway,
So
that's
that's
where
what they use. Okay.
that's where the bacterium came from. And the critical thing to acknowledge that any bacterium that can grow in such conditions has really extreme enzymes. And Taq polymerase is one of them. So if we go back, Taq polymerase is this enzyme right here. You put that in with the other components along with your target, and every time, every cycle, you heat the reaction mixture up to almost boiling, about 95 degrees. Any other DNA polymerase would inactivate, and that would be the end of the reaction. Taq polymerase just tolerates it. It's not active there, but it doesn't denature. Then when you cool it down to 75 degrees, Taq polymerase is a polymerase that makes a copy of DNA. Oh, I'm running out of time, so I won't go over how weird a guy Kerry Mullis was. He was a very strange man. Unfortunately, he died 2 years ago at age 74. But what are the applications of PCR? There are many. In fact, anytime a large amount of DNA is needed, PCR is used. So for example, analysis of clinical specimens for infectious agents like viruses or bacterial diseases, including coronavirus. You hear about this all the time. You hear about testing for coronavirus. The standard test is a PCR reaction. Identification of mutations. So for example, cystic fibrosis, which we've done a lot of study on. Forensics, I've told you about that. Ancient DNA, from any source, bones, fossils, hair, mummified tissues. PCR can be used to amplify DNA that's thousands or tens of thousands years old. And that's how we know that most of us are part Neanderthal. Let me go on. How do we know that? We know it because of PCR. A man named Svante Pabo, and I'll talk a lot more about this when we talk about evolution. But in 2010, Pabo sequenced the genomes of 3 Neanderthals from bone fragments preserved in a cave. And this was just 7 years after the first human genome sequence was completed. So how did Pabo do it? With PCR. From these bone fragments, he did PCR reactions and got as much DNA as he needed to do analyses. And so, for example, he got evidence. One of the genomes in this cave was a red-haired woman. And her DNA sequence indicated that she was an inbred, that her father was a half-brother of her mother, and she was a Neanderthal. Now, present-day humans have some Neanderthal DNA, depending on the ethnicity. So Africans don't have any. And I'll tell you why when we talk about evolution. Europeans, Japanese, Chinese, they all have a little over 2%, Polynesians 4%. I myself have 3.1%, fairly high amount of Neanderthal DNA for a European. So that's the end of PCR. I'm trying to make the point for 2 points. One, how it's done. So you know when you hear about PCR in the news, you know how it's done. And second, what it's used for. Okay, gene therapy. Delivery of nucleic acid into a patient's cells as a drug to treat disease. And the approach here is to treat the cause of disease rather than the symptoms. And it's really done in one of two ways, illustrated in the diagrams here. Either you can add a gene, so you can introduce a gene into a cell that has a mutation, the red dot is a mutation, so that the cell will now make both normal product and mutant product, and the normal product will prevail. Or you can do a recombination event where you introduce a piece of DNA that undergoes recombination with the resident gene, and it replaces the mutant sequence with wild type sequence. So these are the two basic approaches. And just to remind you that CRISPR is the best way to do these things. And I want to see. A number of you, when you made your presentations about cystic fibrosis, and you got to the part about therapies, you mentioned gene therapy and you mentioned CRISPR. And that's not incorrect, but it's limited. So I give you an example here of just how far we've come and how far we have to go in the use of CRISPR to treat cystic fibrosis. So I should say that gene targeting requires dividing cells. Cells in organs that are affected by cystic fibrosis, like the lungs and the pancreas, are not dividing or not very fast. And so the people who did this particular experiment used stem cells that they got from the intestine of a patient with cystic fibrosis. So here's the intestine, and these organoid cultures were taken and grown. The cells were grown in petri dishes from a cystic fibrosis patient. And then these organoids were treated with a protease so that they'd be separate cells. So these are cells in a suspension, and then they were treated with CRISPR. And CRISPR was designed to replace the mutant CFTR gene with a wild type sequence. And then the cells were grown again, and they stayed in culture. And then an assay called forskolin was done to see if this gene replacement had actually worked. And in the upper right here is a diagram of the constructions that were used to replace the mutant gene here in red, replace the mutant gene with the wild type gene. And in the lower right is the assay to see if it worked. Now this is an assay for chloride transport. It's called organoid swelling, but what it reflects is the transport of chloride ions. And what you see is that the controls here at the bottom with a CFTR mutation did not transport chloride. But the blue line and the green line here are cells in which the mutant gene was replaced with wild type. And you can see that they transport chloride very efficiently. So that proves that it can work, but these are cells in culture. They're not treating a human body. The problem with gene therapy now is getting the construct into the cells where it's needed. So you can do it in dividing cells in a test tube. So far, it's been very difficult for any disease to do it in a body that's already developed, a diploid body. And as we talked about earlier, doing this sort of thing in an embryo is forbidden. It's considered unethical at this point in humans. So we are out of time, I'm sorry to say. I want to talk about genetically modified organisms, and I'll do that next time if I can work it into the schedule. But if I can't, we're going to have to skip GMOs this time around. It's something that you should know about. Okay, that's it. Any questions before the end of the period?
When will it be the next quiz, Professor?
Next quiz will be a week from Wednesday. So not this coming Wednesday, but rather November 17th, Wednesday, a week from Wednesday.
Okay. Thank you.
Professor,
Uh-huh.
another thing that you possibly could do is record the GMO information, and then we can just pull it up like the same as you do with this stuff.
That's a good thought. Okay, yeah, I'll try to present it in lecture next time, but if I can see that I'm not going to work it in, I'll do something like what you say. Thanks for that suggestion. Anything else?
No, that will be the last quiz before we go out for Thanksgiving break, and on the 22nd, I think, the last day.
Yeah, let's see. Yeah, the 17th, and then, yeah, the following week, the 22nd and 24th will be Thanksgiving break. And then we don't come back until November 29th, which is the last Monday in November. And then in December, we're very close to the end of the semester.
Okay.
So time's getting.
But you won't be giving us anything over the holiday break, I hope.
I'll say that again.
Will you be giving us any work to do over the Thanksgiving holiday?
No.
Thank you.
No, what I suggest you do is if you want to do something, listen to these videos and prepare yourself for test 3 and for the final exam.
But you'll have those study guides available for that. Okay,
Yeah.
well, hopefully nothing, because, yeah, I got to cook, so I don't even have time for that. Okay, thank you.
All right. Anything else? I'm gonna.
Bye-bye.
Bye.
We're good. Bye.
Bye, everyone.
Bye. See you Wednesday.